To continue chapter 3, Mendelian genetics, we can talk a little bit about how genetic data is interpreted statistically. And one of the really common statistical analyses that we do for genetic data is called a chi-square analysis. So with this chi-square analysis, we're really trying to determine whether or not, at least applied to genetics, whether or not a genetic event occurred due to chance, or what is the probability or the likelihood that a particular genetic event occurred due to chance. When you have a genetic event that it's basically random, right? It was due to random chance, due to a random mutation or some other random fluctuation. Those events are usually pretty rare, and we can look at larger and larger sample sizes in order to get a true idea of, is this event we saw truly a random chance occurrence, or are we starting to see a pattern that might be leading to this genetic outcome? So the two things that we look at when we're looking at using statistical analyses to analyze genetic data are one, independent assortment. Remember, we've talked a lot about the idea that independent assortment can provide genetic diversity. Well, sometimes that independent assortment can also lead to chance genetic outcomes. But again, the difference between different genetic outcomes changes as we increase the sample size. So the larger sample size you have, the more accurate of a prediction you can make about a various genetic outcome. And we can even use that if we start to think about our Mendelian genetics that we talked about on Thursday, the purple and the white flowers, the yellow or the green pea. We have this idea now that our expected outcome is, if we cross two heterozygotes, that we get a 3-to-1 phenotypic ratio. So that's our prediction. But if we had a really small sample size, if we only, let's say we only did 4 or got 4 plants out and they were all purple or they were all white, there's a really good possibility that getting all 4 purple or all 4 white arose just by chance because of randomness and independent assortment, the randomness of which gametes get made and fertilized, etc. As we start to increase our sample size, just like Mendel did with hundreds or thousands of plants, you really start to see what the true pattern is, and that true pattern became that 3-to-1 pattern. So, with a small sample size, you may not get as accurate a result or as reflective of a result as what you should have gotten. So when we're generating our hypotheses about an expected outcome, if let's say we use our purple and white flowers, we create what's called a null hypothesis, that we predict if we do a cross of 2 heterozygous plants that have purple flowers, we expect a ratio of 3-to-1, 3 purple to 1 white as our predicted value. So the null hypothesis is that what we see should fit our prediction. And if there is a difference, that's because of chance. And so we use this statistical analysis to give that some weight or some backbone. So the chi-square test, or the goodness of fit, basically looks at the data that we've received and compares it to what we predicted to see if our null hypothesis actually was correct. And so we're looking at our observed. So what we actually saw, we did the cross, what plants did we actually get, and our expected. So for example, with our 3, let's see here, I can write on the screen. with our 3-to-1 ratio of purple to white, let's say I'm going to breed and get 1,000 offspring. So my expected results out of 1,000 offspring, I would expect 750 to be purple and 250 to be white. That's my expected. Out of the 1,000 plants, let's say I actually observed, just to use a round number, 800 purple and 200 white. So what we're trying to say here is, yes, my observed results are different from my expected results. But is that by chance? Just the random nature of independent assortment of which gametes actually end up being used for fertilization? Or is there something else going on that makes this difference not just because of chance? Is there some other factor that might be playing a role that means my observed results are not because of chance, but because of something else? And so that's what this chi-squared test is designed to do. So here they've done an example with different observed and expected. So here the observed isn't too far off from what we would have expected, right? It looks like it's 10 off in either direction. So the way that you do the chi-square test is you calculate what they call the deviation, basically the difference between your observed and expected. You then square that and you divide it by your expected values. And so basically you're looking at what percent of your expected value did you deviate by. So here, 13, 40, you add it up and you get this constant or this value called the chi-square value. And then you can compare it to a table to get what's called a p-value. And the p-value, people debate about what p-value means all the time. But just to really, really simplify the p-value, the p-value is basically the likelihood or the probability that the event observed was due just because of chance. And so when a p-value is the p-value that we often think about in science, and if you've taken statistics, you've probably talked about it. If p is less than 0.05, we consider that to indicate that our results are likely not because of chance. So that means that there's less than a 5% chance that our results are because of some sort of randomness. Really, that means there's probably an effect going on. But in this case, this p-value is pretty high. So they're saying, there's a 48% chance that the results are not due to randomness. So it's probably this difference that we're seeing here is not significant. It's within what can happen when you're doing the math. So it's important to think about things that way, that we really have to consider not only what our results are, but are they what we expect? If they're different from what we expect, is that significant? Is that significant biologically? All of that. One of the other things you need to know in order to do a chi-square analysis is what's called the degrees of freedom. And you can use this to do a table. And I'm not going to ask you how to find the value on the table, but I might ask you to interpret whether or not the data indicate a significant difference or just a chance difference. And that's because of that p-value. So to calculate the degrees of freedom, you do n, the number of different categories, minus 1. So when you're doing a 3-to-1 ratio, when you have only 2 phenotypes you're looking at, right, purple or white flowers, you have 2 categories, and then you subtract 1, right? So 2 minus 1 is 1, so you have 1 degree of freedom. But if you were comparing A dihybrid cross, where you have 4 possible phenotypic outcomes, 4 phenotypic outcomes, your n is 4 minus 1 equals 3 degrees of freedom. So that's how you calculate the degrees of freedom. And then on this slide, again, they're just trying to show you if your degrees of freedom are this and your chi-square value is that, what is your p-value going to be? And so that tells you whether or not, so everything, basically everything up here, from here up, if the combination of your probability and your chi-square value is up here, that means it's probably due to more than just randomness. If your number is in this lower blue box, right, any differences you observe are most likely due to just random chance. And again, this is the table. So we saw that on the previous slide here, the p-value, right, was 0.48, but what we're interested is also this chi-square value, 0.53. So the chi-square is 0.53, so let's go back here. So the chi-square, okay, we have our, here's our, right here for 1 degree of freedom. So we look here, 1 degree of freedom. Okay. And remember our chi-square value was 0.53. And so this chart here doesn't specifically list 0.53, but it does list 0.46 and 1.64. And so of course 0.53 is going to be somewhere in there, so that we know our chance value is somewhere between 0.5 and 0.2, meaning it's not a significant difference. The last section of this chapter is actually one that I think is really fun. This one is on pedigrees. And so a pedigree is a way that scientists or geneticists track specifically human families to look at patterns of inheritance and to, figure out whether or not genes are dominant or alleles are dominant or recessive, or are they sex linked. So they're really, really fun. Now, when we're looking at a pedigree, pedigrees should follow like standard conventions so that it's easy to interpret no matter where the pedigree came from. Any scientist should be able to interpret it. And so some of the standard conventions for a pedigree are that circles, so any individual represented by a circle is female. Any individual represented by a square is male. Diamonds are individuals of unknown sex. Parents are connected by a single horizontal line. The offspring are a vertical line from the parent. If you have a double line, that means the parents were related. For example, 2 cousins. Other conventions, if the parents had multiples, if they had twins or triplets, you would have diagonal lines that come from a vertical line. They basically are trying to indicate that 2 or more children were born from the same pregnancy. If the twins are identical, they'll connect them with a horizontal line. If there isn't, if they're fraternal, they're not identical, they won't have that connecting line between the diagonal lines. And the last convention is the person who's called the proband. And basically the proband is the person in the family who was first recognized to have the condition. I've seen a lot of pedigrees that have this, but I've also seen a lot that don't. So not every pedigree is going to indicate which person actually brought the family to the attention of medical personnel because of the condition. But if a person has been identified, they'll have that letter P. So again, this is trying to show you some of those standard conventions. Female, male, sex unknown. Any individual that is afflicted with the condition will be shaded, so either a shaded female or a shaded male. Parents who are unrelated will be connected by a single line. If the parents were related, consanguineous, the same blood, usually, we're not talking about siblings, usually cousins, they'll be connected by double lines. The offspring will be listed in birth order. So this family had a male, then two females, then a male. If the offspring were twins, basically right here you would have the first twin come and then the second twin to indicate that they were born at the same time. And as you can see here, fraternal twins will have no connecting line. Identical twins will have a connecting line. And identical twins have to be the same sex. Fraternal twins can either be the same sex or they can be opposite sexes. If a family has a lot of children and many of them are not affected and their children are not affected, you might just see, well, they had 4 boys, 4 girls, and then it might pop out, the 9th child if that child was affected. The proband, if identified, will look like this. An individual who is deceased will sometimes have a circle through them, whether or not they were affected. So this is actually an unaffected female who has been deceased. Individuals that are known to be carriers of a trait but do not express the trait will have a dot because they're heterozygous carriers, and then each generation is labeled with a Roman numeral. So to actually look at a pedigree, what we're looking at in the top is a pedigree for a family that is afflicted with a recessive, autosomal recessive trait. So remember, autosomal means non-sex chromosome. So when a trait is recessive, right, like even our purple and white flowers, this might be the purple flower, right? This would be the white flower. You often see that it skips a generation. So we see that none of the individuals in generation 2 are afflicted. So that's a very, very good sign that you're looking at a recessive trait. Now, it doesn't have to mean that, but it usually does. And sometimes you might even still see affected individuals, right? If you saw an affected individual in the 2nd generation, that would probably mean that mom was recessive, right? So recessive traits often will skip generations, and then you see, okay, well, actually this family, so these 2 parents, right, had 6 children. These are identical twins. So they had a male, then a female who has the genetic condition, then a male who has the genetic condition, right, and then they're unaffected twins. So what we have to know now is that, well, neither of these individuals, neither individual 3 nor individual 4, have the genetic condition, but they have children who do. So we know that these have to be heterozygous individuals to have these 2 afflicted children. So in this family that married into the original family, there is at least one of these persons is a carrier for the condition. Now, when we look at this pedigree over here, we're now seeing a dominant allele. And so the dominant trait will almost always appear in every generation. So this means, like, this parent here, right, the mother is actually homozygous recessive, but the father is probably heterozygous because some of his children, right, are unaffected. But some of his children are. And you can see that, again, right, you have a number of offspring that actually express the trait. And if this was a recessive trait, we likely would not see it in generation 2, and we wouldn't have as many individuals likely in generation 3 that had it. So pedigrees are really interesting, and we will look at them a little bit more, especially when we start talking about sex-linked conditions. And so pedigrees, again, have been very useful in human genetic studies to look at a number of different conditions, some of which are recessive and some of which are dominant.